Hello, everyone. Welcome to Brain Blitz Audios. In this episode of NEET preparation, we'll be dealing with previous year questions of biology in medical entrance examinations in India. The chapter that we'll be dealing with is Biomolecules. This is the 9th chapter in the grade 11 syllabus for CBSE. Remember that we've done an earlier episode on biomolecules, and we've discussed some questions. I'll provide the same link in the description down below. Now, in this episode, the most important questions are in the end, so be sure to watch the whole video and get all of the questions. Let's begin, shall we? So, our first question. The Michaelis constant, Km, is equal to option A, K1 divided by K2 minus K3. Option B, K2 plus K3 by K1. Option C, K2 minus K3 by K1. And option D, K1 times K2 divided by K3. So if this is going over your head, let's write down the equation. So Michaelis constant is the constant value where half of Vmax is achieved. So this is the substrate concentration at half of Vmax. That is maximum velocity formed by the enzyme of the reaction. So using an enzyme, what we'll get is E plus S changes to ES, which changes to E plus P. So the forward reaction is K1, the backward reaction is K2, and the final reaction is K3. And so, therefore, we'll write Michaelis constant as the constant of the equations from the enzyme substrate complex, that is K2 plus K3, divided by the constant of the equation, the reaction that leads to the enzyme substrate complex, so that is K1. So K2 plus K3 divided by K1 is Michaelis constant. And therefore, we find that option B is the right answer. So remember that it is K2 plus K3 because we are considering both the equations, the backward equation to enzyme plus substrate and the forward equation to enzyme plus product. Next question. Alpha-keratin is a protein present in A, blood, B, skin, C, lymph, for eggs. Keratin is a dead protein found in the body. Let's look at our following options. Option D, eggs. Eggs have the protein albumin and not alpha-keratin. So option D is incorrect. Option C, lymph. Lymph contains many proteins, the most prominent of which is laminin. So option C is incorrect. What about option A, blood? Blood is primarily composed of hemoglobin, the protein which contains iron that transfers oxygen in the body and which also gives blood its red color. So option A is incorrect. The right answer is option B, skin. The outer layer of skin composed of dead cells, which then shed off as dandruff, and that is made up of alpha-keratin. The protein alpha-keratin makes up the outer layers of the skin. So the right answer is option B. Next question. Which one of the following statements regarding starch and cellulose is not correct? So we're asked to find out which of the statements is not correct. Let's look at each of our statements. Option A, both of them are of plant origin. Cellulose is found in plants, and the starch is what we get out from the grains, that is the seeds of the plant. So both starch and cellulose are of plant origin. So the statement is true, which makes the option false, because our question is to find out which of them is not correct. Option B, both of them are polymers. It is true, starch and cellulose are polymers of glucose. So option B is also true, which makes it incorrect. Now, since starch and cellulose are made up of glucose molecules, we can say that option D is true, which makes it incorrect. The right answer is option C. Both of them give color with iodine. This is incorrect because cellulose does not give color with iodine. And the reason why is that starch is a branched polymer, which is soluble in water and reacts with iodine, and cellulose is a linear polymer. Both polymers also have different types of cellulose. Starch contains alpha-D glucose, and cellulose contains beta-D glucose. So therefore, we can say that option C is the false statement, which makes it correct. Next question. Which of the following type of enzyme is not matched correctly with the molecule that it breaks down? We need to find the incorrect matchup in this scenario. So we have amylase matched up with starch, lipase matched up with starch, protease matched up with proteins, and disaccharidase matched up with sugars. Let's look at each of our options. Option D, disaccharidase. So disaccharidase is an enzyme which catalyzes disaccharides, which are sugars. So therefore option D is true, which makes it incorrect. Option C, protease catalyzes proteins. So the protease is an enzyme that breaks down proteins. An example of that is pepsin and trypsin. So therefore, C is true, which makes it incorrect. Now, if you look at both of our options, A and B, both enzymes, amylase and lipase, are set to break down starch. So we know that one of these has to be incorrect. Which of them is incorrect? The one which is not matched correctly and which is our right option is option B. The reason lipase breaks down fats and not starch. The enzyme that breaks down starch is amylase. So amylase breaking down starch is a true matchup, which makes it incorrect. But lipase catalyzes fats, it breaks down fats, so therefore it is incorrect. It is false. So that option is the right option because we're asked to find out which of them is not matched correctly. Next question. The diagram illustrates energy changes in an enzyme-controlled reaction. Which of the following represents the lowering of activation energy? X, Y, Z, Z minus Y. So Z represents the activation energy of uncatalyzed reaction. And as you can see, it has a higher place in the graph. It has a higher difference. So that means the energy of activation in an uncatalyzed reaction is very high. Y represents the activation energy of a catalyzed reaction. So the reason why enzymes are so effective in catalyzing reactions is that they lower the activation energy. So as you can see, Y is lower than Z. So since Y is less than Z, we can write the lowering of activation energy, that is the difference between the activation energies as Z minus Y. So Z minus Y is option D, so it is correct. Z only represents the EA of uncatalyzed reaction, so it's incorrect. Y only represents the EA of catalyzed reaction, so it's incorrect. And X gives us the energy of the products, which is also incorrect. The right answer for which of the following representing the lowering of the activation energy is Z minus Y. Next question. Which one of the following is a non-reducing carbohydrate? Maltose, sucrose, lactose, ribose 5-phosphate. Let's look at each of our options. Option A, maltose. The structure of maltose has a free aldehyde, and aldehyde is reducing, so that makes option A incorrect. Option C, lactose, also has a free aldehyde group in its structure, so therefore option C is also incorrect. What about option D, ribose 5-phosphate? Ribose 5-phosphate is instrumental in creating NADPH, which is a reducing equivalent. It undergoes reduction. So therefore, option D is also incorrect. The right answer is option B, sucrose, a disaccharide, which has no reducing group, no ketones, no aldehydes. So therefore, option B, sucrose, is the non-reducing carbohydrate. Next question. The Km value of the enzyme is the value of the substrate concentration at which the reaction reaches 2. 0, 2 Vmax, half Vmax, 1/4 Vmax. So this is basically a definition question. So if we know the definition of Km, which is the Michaelis constant, we would know which is the substrate concentration where the reaction reaches. So let's look at our options right now. Option A, 0. So when the value of the substrate concentration at which the enzyme reaches to 0 will also be 0. So what that means is that no reaction takes place. So option A is incorrect. Option B, 2 Vmax. This scenario is not possible because the highest velocity that an enzyme can go through, the highest rate of reaction that an enzyme can reach, is known as Vmax. Therefore, 2 Vmax is not possible. Option D, 1/4 Vmax. This quantity is not defined, so it's incorrect. The right answer is option C, half Vmax. In the graph, whenever an enzyme reaches Vmax, this point, that is the half Vmax, and the substrate that is reaching here, so you see the substrate concentration when the rate reaches half Vmax is known as Michaelis constant, that is Km. Next question. The following diagrams represent the nitrogenous bases of nucleic acid molecules. Identify the correct combination. So the 5 bases are adenine, guanine, cytosine, thymine, and uracil. And we have given them in different combinations. We need to find out which of these combinations is correct. So when it comes to nitrogenous bases, there are a couple of things that you need to know. Nitrogenous bases are divided into purines and pyrimidines. Purines have 2 rings in their structure. So therefore, B and D are purines. And among the purines, adenine has no oxygen group. and guanine has an oxygen group. So if you look at our diagram, we find that diagram B has no oxygen in its structure. So therefore, we can safely conclude that B is adenine. And when we look at our options, A, B, C, D, we find that only option A contains B as adenine. So therefore, we can safely conclude without doing much deliberation that option A is the right answer. And when you check it out, you find that D is guanine, which is correct. A is uracil, which is correct because it doesn't have a methyl group attached here, only a hydrogen. And thymine is C because it has a methyl group unlike uracil. And cytosine, which has an NH2 at the top. So that makes option A the right option. Remember, we can also use this by comparing uracil and thymine, but that would not be as effective because there are 3 options with A as uracil. So we compared adenine, and using the fact that adenine does not have an oxygenous structure, we found out that option A is the right option. Next question. The given graph shows the effect of substrate concentration on the rate of reaction of the enzyme green gram phosphatase. Now we need to find out what does this graph indicate, and our options are: the rate of enzyme reaction is directly proportional to the substrate concentration, presence of an enzyme inhibitor in the reaction mixture, formation of an enzyme substrate complex. At higher substrate concentration, the pH increases. So when we're looking at this question, one thing is to be clear. The question is specific to the given enzyme, green gram phosphatase, and the given graph. So when we look at our options, we find that the rate of enzyme reaction is directly proportional to the substrate concentration would be incorrect because this is a general observation among all graphs of the same kind as this one. So when we are representing the effect of substrate concentration on the rate of reaction, we can do this to any kind of enzyme, and you can still get the relation that velocity is directly proportional to substrate concentration. And formation of an enzyme substrate complex. Option C is also a general observation for all types of enzyme catalyzation, so therefore it is incorrect. Option D is also a general observation. So at higher substrate concentration, the pH increases. The right answer is option B, presence of an enzyme inhibitor in the reaction mixture. In a normal graph, the graph goes up and then it stays constant at a particular value. That value is Vmax. And half of Vmax, which we can represent as Vmax by 2, gives you the Michaelis constant, Km. But over here, you find that this graph never reaches a constant state. It starts to decrease. And when does the velocity decrease? Velocity decreases when there is an inhibitor present. So that is something that's unique to this graph. So therefore, we find that option B, presence of an enzyme inhibitor in the reaction mixture, is the right answer. Next question. Inorganic catalysts work efficiently at dash temperature and dash pressure. We should find it whether they're at high temperature, low pressure, low temperature, low pressure, low temperature, high pressure, or high temperature and high pressure. So Let's take an example of an inorganic catalyst, MgSO4. In order for this inorganic catalyst to work, it needs high pressure and high temperature. So therefore, using this example, we can conclude that option D, inorganic catalysts work efficiently at high temperature and high pressure, is the right answer. So all of the others are incorrect. Option B in particular is for enzymes and not inorganic catalysts. Now, we've reached our final question. Refer the given structure of adenyllic acid. In this, identify the bond A given here. We need to find out whether A is a glycosidic bond, a phosphate bond, an ester bond, or an ionic bond. So, for those of you who don't know, adenyllic acid is a nucleotide. And a nucleotide contains a base and a sugar and phosphate. So we have a nitrogenous base, we have a sugar, and we have a phosphate here. So let's look at each of our options. Option D, ionic bond. Ionic bonds are found in this carbon ring. So therefore, option D is incorrect. Option B, phosphate bond. In order to find out which of these is a phosphate bond, we need to know that phosphorus goes with oxygen in order to get phosphate. So therefore, the phosphate bond is the double bond right here. Next, glycosidic bond. The glycosidic bond occurs here between the sugar and the phosphate, like this. So therefore, the ester bond is the bond that occurs at A. That is the main link between the sugar and the phosphate. The glycosidic bond links another part of this nucleotide. So therefore, option C, phosphate bound to the pentose sugar to form a nucleotide, is at A. So option C is the right option. Let's draw that once again. The ester bond is right here, because you see there's an OCH2 present. So this was the penultimate question. And now to our final question. Nucleotides are building blocks of nucleic acid. Each nucleotide is a composite molecule formed by sugar phosphate, base sugar phosphate N, base sugar OH, base sugar phosphate. Sugar phosphate is expressed as it is. It has no other classification such as nucleotide or nucleoside. So option D is incorrect. Option C, base sugar phosphate raised to N. This is a polymer, and the polymer in question would be nucleic acids. We are asked to find out the composite molecule of a nucleotide. So therefore, option C is incorrect. Option B, base sugar OH. This type of compound does not exist in DNA and RNA, the nucleic acids. Therefore, the right answer is option A, base sugar phosphate. Nucleotides contain a nitrogenous base such as adenine, guanine, cytosine, thymine, or uracil, and a sugar that is ribose or deoxyribose, and a phosphate in the middle. So therefore, option A is the right option. We know that option C also contains the same, but since the bracket raised to N refers to a polymer, and the polymer here is nucleic acids and not nucleotides, this option is wrong. And so therefore, we've completed this episode of NEET preparation. There are a lot of questions in NEET about various topics, and it's impossible to discuss them all in a single video. So therefore, we've attached some links in the description down below. Be sure to check them out after watching this video completely. And for accessing more of our useful and interesting content at Brain Blitz Audios, please subscribe to our channel. We'll be back with more useful and interesting content such as learn awards, mind maps, NEET preparation, CBSE board exams, etc. So until the next episode, take care, stay safe, ta-ta for now.